Hola, Team Bio. Okay, so today's screencast is on Mendelian genetics. So far, you know a little bit about the ways in which chromosomes are constructed and how genes are found on chromosomes and how different alleles are different variations of the same gene. But we haven't really talked about inheritance and how genotype, meaning the DNA sequence you have, the different alleles that you have, relate to phenotype, which is what you present as. So you have all the cells in your body contain the same set of chromosomes and the same genes and alleles, and then that manifests itself as physical characteristics, either traits or behaviors. And so we're going to talk a little bit today about the way in which genes are inherited and passed down and how genotype relates to phenotype. So that all starts with Mendelian genetics is what the topic that covers that. Oh, God. Monday. Sorry, I'm a little bit all over the place. Okay, so Mendelian genetics was invented by a guy named Gregor Mendel. He was a monk living in, I think, somewhere in Austria, actually. And he was doing an experiment with pea plants. And as Mendel was doing his experiments, the common belief of the time is that traits are inherited as some sort of type of blending inheritance. Blending inheritance. So, for example, if, you do have a mother and a father, and for the purposes of this example, let's say that your mom is really tall and your dad is really short, and they have a baby, which is you, and you will probably be somewhere in the middle of them, somewhere between your mom and your dad. So this is the idea that when people pass on their genetic material, that there's some sort of blending that happens and you meet somewhere in the middle, which is not a crazy thing to believe if you look at people. Often kids seem like a combination of both their parents. But Mendel found out, in his experiments on peas, found out that blending inheritance is really wrong, and that instead of genes kind of coming together and blending it to meet somewhere in the middle, that genes can be passed down in discrete units. So I'm actually going to select all of this and delete it because it was wrong, and say that genes passed, not bound, down in discrete units. Okay, so I'll give you an example of this. So Mendel was doing, as I think I said before, he was doing experiments on peas. And so he had a bunch of different varieties of peas. And if he found that if he took a pea that was round, and let's say it's true breeding round, so its parents were round and the parents before it were round, and so on and so forth. So it comes from a long line of round peas. And he crossed that with a pea that was wrinkled. And again, this is a true breeding wrinkled plant. It came from a long generation of parents that were also wrinkled. I don't like that it's not fully colored in. Okay, there we go. So he crossed these two together, meaning he mated them, and they had babies. He found that in this case, where he crossed these two plants, and they had offspring, that every single one of their offspring was a round plant. Oh, you know what? This is silly. I should just say cut. Oop, nope. I'm just going to copy these. Paste. Aha. Paste. I don't know if I'm saving a lot of time here, actually. Paste. Paste. Okay, so across these two plants, and every single one of the progeny, the offspring, are round. Now, I'm gonna label these generations so we don't get confused. This first generation, the round and the wrinkled plant, we're gonna call the P generation for parental. And this next generation we're gonna call the F1 generation. Actually, I don't know what F stands for. Maybe filial? I'm not sure. Anyway, so we have the P generation and we have the F1 generation. Okay, so, and all the F1s are round. Now, here's where it gets interesting. Mendel found that if he took two of these F1 guys and crossed them together, I'm going to keep a similar drawing schematic as before. So he crossed these two F1 guys together, and they had offspring. This is where it gets exciting. He found that they produced a round baby, and then another round baby, and then another round baby, and then a wrinkled baby. Ooh. Okay, so this was a little bit confusing to him. Okay, and 1 second, back up. This is called, right here, this is called the F2 generation. So in the F2 generation, you see the reemergence of this wrinkled P. So this kind of confused Mendel, but he saw this ratio again and again and again and again. This 3 round to 1 wrinkled over and over and over again. And it took him a while to kind of figure out what was going on behind the scenes here. But ultimately, what he discovered, or what he hypothesized, was that every parent comes with 2 copies of a gene. And you all know this because you've seen that we all inherit 1 chromosome from our mom and 1 chromosome from our dad. So we call these 2 copies of 1 chromosome homologous chromosomes. So in this example, in the P example, if we scoot up here to the top again, this round P right here was a true breeding round P, meaning all its parents were round, so it was big R, big R. It's not a great R, but there you go. And this wrinkled P was little r, little r. So this is traditional, typical nomenclature. We denote the capital letter of whatever is the dominant trait, and the little lowercase letter is whatever is the recessive trait. And we know that round is dominant to wrinkled because in all of our offspring, we saw round P's. So you can kind of depict and you can predict what type of offspring you are going to see in the F1 generation by creating what's called a Punnett square. And at the top of the Punnett square, you put the alleles that the father has, and on the side, you put the alleles that the mother has, so the gametes that they can produce. So the father can only produce gametes that are big R because both his chromosomes contain the big R allele on his round or wrinkled, their P's body shape gene. And the mother can only create gametes that are little r because she contains 2 copies of the little r allele. So when you put those together, you see that all the offspring will inherit a big R copy of the allele, round, and a little r copy of the allele, wrinkled. So they are all called what is called heterozygous. This is in comparison to their parents, which were both homozygous. So hetero meaning different, and homo meaning the same. So the parents had the same alleles, big R and big R, little r and little r, and the children have different alleles, one big, one little. But you can see quite clearly here that the big R is dominant over the little r because all the offspring had the round phenotype, even though they had one allele for the wrinkled genotype. Okay, so that's how we explain what happens in the F1 generation. But how about the F2 generation? Well, we can create the same type of Punnett square to the side right here. And if we look at these two parents right here, we see they're both heterozygous, which means they both have a big R gene that they can pass down, and they also have a little r gene that they can pass down. So both parents are going to look like this. And then, when we do out our Punnett square, we see that these are the genotypic ratios we're going to expect to see in the F2 generation. We're going to expect to see around P, around P, around P, and a wrinkled P. So, for every 4 offspring, this is the ratio that we'd expect to see. Now, you know a little bit about probability. If you only have 4 offspring, you might not see this ratio, but as more and more offspring are created, you expect to be closer to this 3-to-1 ratio. Oh, and I guess I should annotate this up here. All the offspring here are round. So, again, looking at the P's that we have here, we see that 2 of the offspring are heterozygous, HET, and 2 are homozygous. However, if we were to look at the offspring that we've created here, the only genotype that we can definitively state here is little r, little r. We know this must be a little r, round plant because it is recessive. However, this could be big R, or big R, little r. We don't know. Likewise, big R, little r. We don't know which one of our round P's are true breeding and which ones are heterozygous. Okay, so that, I think, is it for this screencast. Just a taste of Mendelian genetics. We're going to be working on more of these types of genetics problems in class. Probably not tomorrow, but a few days from now. So that is it. If you have questions, I'm happy to answer them, and au revoir.